Okay, so chapter 10 is going to focus on the third of three types of energy producing processes called photosynthesis. Talked about photosynthesis is performed not just by autotrophs, organisms that are capable of synthesizing their own nutrients, utilizing inorganic compounds, but these are organisms that are photoautotrophs. In other words, they do use inorganic compounds, as you'll come to see, carbon dioxide and water, but they also have the ability to capture, absorb, or harness a type of kinetic energy called light energy. Now, we're going to focus on plants in this chapter, but this figure is just to let you know plants aren't the only organisms capable, okay, of photosynthesis. There are bacteria capable of photosynthesis, such as cyanobacteria, and there are protists, such as brown algae, or some people call it giant kelp in the case of this picture, as well as euglena. Okay, they're also capable of photosynthesis. Now, bacteria down here, cyanobacteria, I'm not going to test you on it. They don't have organelles we're going to focus on that you find in these other three. Okay, but they do have molecules of chlorophyll, and they do have a key enzyme needed to perform photosynthesis. Okay, so we talked about fermentation and cellular respiration. And cellular respiration was an aerobic process, energy-producing process that required glucose and oxygen. But there's a cyclical relationship between cellular respiration and photosynthesis. Photosynthesis falls under anaerobic metabolism. It doesn't require O2 as a reactant, okay, to catalyze a series of chemical reactions or a group of chemical reactions catalyzed by a group of enzymes that make up photosynthesis. Instead, the gas that's needed is CO2. But CO2 and water, these are two of 5 types of inorganic compounds that many autotrophs can utilize to help produce energy. Except in the case of photoautotrophs, not only do they use these two inorganic compounds, water and carbon dioxide, they have molecules called chlorophyll in them that allow them to we often say these molecules of chlorophyll absorb light energy, okay, and that allows the plant or any organism to perform photosynthesis. But as I'll tell you, the better way to describe absorb is we'll talk about how due to the fact that these light absorbing molecules called chlorophyll, basically what's happening is these molecules of chlorophyll are taking this type of kinetic energy and transferring it over to themselves so that they can help with this process of photosynthesis. Now, the purpose of photosynthesis is not to synthesize ATP. The purpose of photosynthesis is to synthesize the organic compound glucose, as we'll talk about. Now, there will be a waste product, which is O2. So in this case, O2 is a waste product, but that's fine. These two are needed for cellular respiration. So this is just highlighting the similarities. Okay, here, the exact similarities. What are product photosynthesis are reactants needed for cellular respiration. What are reactants for photosynthesis? You can see 2 of the 3 reactants are actually 2 or 3 products for photosynthesis. So these 2 equations have a lot in common with each other. Now what we're going to do is this right here for photosynthesis. The formula I'm giving you, okay, it's like cellular respirations. It's not necessary, or how to say exactly, a chemical reaction such as 2 molecules of atmospheric oxygen plus 1 molecule of 2 molecules of atmospheric hydrogen plus one molecule of atmospheric oxygen produces 2 molecules of water. It's not anything like that. It's more of a summary, just like before. So I'll dissect this formula just like I did for cellular respiration, except I'll dissect it a little more neatly. Okay, these are the initial reactants that are needed. These are final products, but there are products in between this point and this point we'll talk about. Okay, so as I said, there are bacteria called cyanobacteria that are protists such as euglena, or protists such as brown algae, which is actually a multicellular organism, most popular type, giant kelp. They are capable of a process called photosynthesis, but we're going to focus on plants. Okay, when you look at plants, the primary part of the plant that stands out are the leaves, which are green colored. Okay, what they've done is they've taken a small piece, excuse me, they've taken a small piece of the plant leaf and they've magnified it. Okay, so this is what the leaf would look like under a microscope, say 400x total magnification. Looks like you're looking almost at the side of a brick wall, except it's green, the bricks. What you're looking at here is the white color that you see here, or the whitish color that you see here. That's the cell wall okay, or cell walls of these two plant cells meeting up here, cell walls of these two plant cells meeting up here. But what really stands out are these little green-looking bead-like structures. These little green-looking bead-like structures, these are organelles, membranous organelles called chloroplasts. So in this next figure, what they do is they take a small section of the leaf, the part of the plant that's green, except you're taking a lateral view. You're looking at the leaf from the side. And they're showing you this is what it could look like if we drew a nice, neat diagram. When you look at the anatomy of a leaf from the side, from a lateral view, or as they say, a cross section here, okay, plant leaves have an epidermis just like we have an epidermis, the superficial part of our skin. And they're showing you here and here. And they're trying to show you by these little, rectangular shaped structures that the epidermis of plant leaf is made up of cells, but these cells aren't green. It's what these cells that make up the epidermis of the plant leaf are protecting. That's where the green color is coming from. We call this layer here is called the mesophyll, but what I want you to know going into the test is these cells here that they're showing you that appear green, these are called mesophyll cells. And if you look here at the figure down here, they're showing you it's these mesophyll cells that have these green beads inside of them. Therefore, it's your mesophyll cells that are packed full of these organelles called chloroplasts, of which they're showing you one here. Now to backtrack, going back to the leaf, okay, they're showing you that a leaf has small pores or openings that are required for gas exchange. Okay, these are called stomata. Okay, so it's showing you this is plural instead of stoma for singular, but no, that's stomata going into the test. So what scientists discovered is that these epidermal layers of a plant leaf, they actually have small pores. Now, I'm not going to test you on it, but these little brownish colored guys, they're actually cells that can either close up the pore or the opening, or they can pull apart, creating that pore or opening called a stomata. The function of the stomata is gas exchange. It allows CO2 to enter the plant leaf so it can reach the mesophyll cells, which need it for photosynthesis, but it also allows the O2, the excess O2 that the plant doesn't need, to exit this stomata. Okay, now plants do need some of that O2 because they also have mitochondria that can perform cellular respiration, but because of photosynthesis, they have more than enough of what O2 than what they need, so they need to get rid of some of this. CO2 is important, everybody. Without carbon dioxide, photosynthesis will not occur in the end, and I'll talk about this. It's a process called carbon fixation. Okay. Now, as I said, these mesophyll cells, okay, are the part of the leaf that are actually give the leaf its green color because they're packed full of these little bead-shaped structures called chloroplasts. And it's showing you a chloroplast right here, but I'm going to use a figure that's a little bit larger. This is an electron micrograph, or a TEM, transmission electron micrograph of a chloroplast, just to show you, okay, they're not that cut and dry, not that neat and easy to study. So we're going to use this, but it's just showing you a comparison up here. Well, I said a chloroplast is a membranous organelle. If you recall, I said back in chapter 5 that membranous organelles are enclosed structures with membranes made of the same compound as the cell membrane. Okay. The chloroplast, initially, when you look at the anatomy of the chloroplast, starting from the outside, working your way in, it actually begins off a lot like the mitochondria. Just like the mitochondria, the chloroplast has a double membrane. It has a membrane and it has an inner membrane. Okay. These two membranes here are separated by a space, the intermembrane space. So the terminology parallels that of the mitochondria. I mean, it starts off the same. The overall shapes may not be the same, but it starts out the same. Okay, the great thing about photosynthesis, when you go over it as far as location goes, is that unlike cellular respiration, where glycolysis occurs in the cytosol, and then the last two stages, citric acid cycle and oxidative phosphorylation, occur in the mitochondria, the entire process of photosynthesis occurs within the inner membrane. Okay, so when we talk about photosynthesis, all of it occurs within the inner membrane. Now, when we go inside or within the inner membrane, what we first encounter is a solution. Okay, it's often described as a very viscous solution, and I'll use my abbreviation for solution, which is SOL''N. Okay, this is stroma, so don't get it confused with the pore. It's called stomata. Stroma has an R in it. Stomata doesn't. Okay, that's the simplest way I can tell it to you. Okay, but stroma is a solution, and as we're going to talk about, photosynthesis, okay, is made up of or consists of two stages. The second stage is going to happen in this solution, the stroma. This stroma, which you really can't see, they're just pointing as if they're pointing to the inside of the inner membrane, but they're actually pointing to what would be a viscous type solution. This solution surrounds, okay, green-like discolored, what they actually call organelles, okay, green-like discolored structures. These green-like discolored structures, they're called thylakoids plural or just thylakoid for one. If you notice, these little disc-like structures have a green color to them. As I'll show you, the thylakoid, okay, they sometimes refer to these as organelles also because they have what you call a lipid bilayer forming the membranes of these thylakoids, and embedded in their lipid membranes of these thylakoids are molecules of chlorophyll. So technically speaking, what gives a plant leaf its color? It's not just the chloroplast, it's the chlorophyll molecules inserted into the membrane of these structures called thylakoids. Now they just point to one of them. We're going to talk about how of the two stages that photosynthesis consists of, I'm already tripping over my tongue, of the two stages that photosynthesis consists of, the first one happens here in the membrane of the thylakoid. Okay, so these are two key areas. These two parts, the thylakoid with a solution called stroma surrounding them, all located within the inner membrane, and their photosynthesis will occur. Stage one here, stage two here. Okay, chlorophyll itself, as I said, molecules of chlorophyll will be embedded into the membrane of these structures called thylakoids. Well, this is what chlorophyll looks like. Okay. Now listen carefully. In your textbook, and I forgot to get rid of it in your notes, there are two types of chlorophyll, in addition to the fact that there's more than just these two light-absorbing pigments that exist. But I just want you to know it as chlorophyll going into the test. Okay, so going into the test, don't worry about whether it's chlorophyll A or chlorophyll B. Just know chlorophyll. But this is showing you what chlorophyll looks like. The highlighted area here with the background, this is representing the part of the chlorophyll molecule, okay, that's specifically responsible for absorbing or capturing light energy. It's also the area that's responsible for that's attributed to that green color that we get. But chlorophyll, we call it a light absorbing pigment, but I like to describe it this way. Chlorophyll absorbs, or more specifically, captures light energy and puts it to work. Okay? Because if you think about something, if you're talking about absorbing something, it's like putting a dry sponge in a puddle of water. It's going to absorb the water and trap it in the sponge. But that's not exact for chlorophyll. Let me jump ahead here. You got to think of it more like pool. You got to think of light, okay, as being the cue ball. Think of chlorophyll molecules as being the 8-ball. Okay, now first of all, for light, okay, light is made up of light particles called photons. Okay, so again, light is made up of light particles called photons. These photons move at different rates and speeds. Collectively, that creates what we call white light. And that's what this is showing you here. Okay, now I'm not going to test you on specifics here for wavelengths. This is just to let you know. These light particles called photons, they move at different rates and speeds, which correspond to our color spectrum. However, when you collectively take all these photons moving at all their different rates and speeds together, what we see with the naked eye is just white light. Okay? Chlorophyll molecules pick up on certain photons moving at certain rates and speed. Okay, and it's like a cue ball hitting the 8-ball. Recall, light energy is a type of kinetic energy. So what we have is kinetic energy for the movement of photon particles. Okay, and when they strike the chlorophyll molecule, that in turn, in a manner of speaking, excites or causes the movement of the chlorophyll molecule. So kinetic energy here from light transferred to the chlorophyll molecule. Okay, so kinetic energy here, transferring kinetic energy here. Now, I'm not going to test you on this, but why does it appear green? Why does chlorophyll give organisms their green color? Because those light particles called photons, the rate and speed at which they're moving at, chlorophyll cannot capture or absorb the energy from those molecules. They either bounce off or they go right on through. So that's a Jeopardy question thing there. Okay. Okay. What we're going to do now is go into photosynthesis. And what I'm going to do is I'm going to go up through the first stage, which you're going to come to know as a light reaction. And then Thursday, I'll go through the second stage so I can break this up some. Okay, so photosynthesis requires the inorganic compounds, carbon dioxide and water, 6 molecules of each, plus light energy in the form of photons. Sometimes I'll put it up here, sometimes I put it down here to let you know that for the overall process of photosynthesis, these are our reactants. This is what we need to perform photosynthesis. But as I told you before, these energy producing processes that we went over in chapter 9 and we're going over here in chapter 10, they all consist of a series of chemical reactions catalyzed by a group of enzymes. Therefore, as we go through this, I will talk to you about how carbon dioxide and H2O, water, well, that didn't come out, and water here, they're actually substrates. All substrates are reactants, but not all reactants are substrates. Okay? So for the overall process of photosynthesis, we need these 3 reactants. 2 of them are substrates. The final products of photosynthesis are what we want, which is glucose. And then we also get a waste product. So these are our products. These are the final products. And I'll show you in a sec on the next figure how they rearrange this photo and apply it to the 2 stages that make up photosynthesis. Now again, glucose is the final product. It's the purpose of photosynthesis. Okay, glucose is not only a nutrient that can be used to create other organic compounds, it also is a source of chemical energy. Okay, so the plant has 2 options with what it wants to do with this. O2 is a waste product in a photosynthesis. Like I said, the plant produces more than enough O2 than it needs, so some of it needs to be released through that stomata, that pore in the plant leaves. Some of it might be used for cellular respiration. So if you look at this formula now, this overall formula, including the light, and now we go over here. Photosynthesis consists of 2 stages. Okay, the first stage is called the light reaction or reactions. Okay, if you look at this first stage, it's in the name. What does it require? Light or photons. It also requires water. So of the 3 reactants listed across the top here that you need for photosynthesis, 2 of those 3, okay, are needed for the light reaction. And I already said that H2O in this case isn't just a reactant, it's a substrate. I'm not talking because I'm focusing. All right, so it's a substrate. Okay, now if you look at this light reaction, notice they're showing you a final product, one of 2 final products that's produced, which is O2. Okay, O2, like I said before, it's just the waste product, but I'm just going to write product here. So if you look across the top, they're giving you the 3 reactants needed for photosynthesis. If you look across the bottom of this, okay, you have our final 2 products. These 3 are going into the chloroplast across the top. They're going into our chloroplast. These 2 final products are coming out of our chloroplast. However, if you look within, they're showing you a little more right here, and right here is going on. So in your notes, what I do is I dissect this formula for photosynthesis and rearrange it some, or rewrite it in regards to what are the specific reactants needed for the light reaction, and what are the actual products, and then I do the same thing here. So this is stage one, the light reaction. The reactants we need, one of them serves as a substrate, final product we get. However, the light reaction produces two other products that we'll talk about. ATP, and then it shows you here NADPH, but this is an electron carrier written in its reduced form. So let me ask you all a question then. If I'm telling you NADPH is an electron carrier written in its reduced form, what do I mean? It's a third type of electron carrier. You had to know for two for cellular respiration. We talked about oxidized and reduced. If you're doing your studying, you should understand what my question is. So what do I mean by reduced? Yeah, it's gained electrons. And how many electrons for cellular respiration, NADH and FADH2, how many electrons could they gain for cellular respiration? Two, right? Okay. Same thing for NADPH. It's a different electron carrier. It's a third type, and you only find it in photosynthesis, okay, in this class, but it carries two electrons. So just this, everything I'm circling right now has to do with the first of two stages. Okay, light reaction requires these two of three reactants, except H2O is a substrate. The light reaction produces one or two final products, O2, in relation to the formula I gave you at the beginning. However, it actually produces a total of three products, but these two are not final products. Okay, these two are products of the light reaction like O2, except these two serve as chemical energy needed to power the Calvin cycle. So the actual purpose of the light reaction is to produce a stored form of energy called ATP and to transfer electrons that are from water, transfer those electrons with the electron carrier in NADPH. And these two, they will be transported over to the Calvin cycle because these are two sources of chemical energy needed to power the Calvin cycle. Okay, so I'll show you again how it's rewritten. Now, the second stage, and I'm saying this once and I'm saying it again, okay, so don't panic, we'll go over it in more detail. The second stage is the Calvin cycle. Okay. If you look here, of the three reactants I gave you for that formula for photosynthesis, this is where carbon dioxide comes in. Okay, but for Calvin cycle to occur, we not only need the reactant CO2, which is actually a substrate, but we actually need a total of three reactants for the Calvin cycle to occur. We need carbon dioxide. It's a substrate, but I just told y'all before, we also need ATP and NADPH. So in your notes, I had to edit your notes because I left it out and I forgot about it, but I put in, how would we rewrite the formula for photosynthesis in a way that would only apply to the Calvin cycle? Well, we would rewrite it CO2 plus ATP plus NADPH, not 6CO2, but ATP plus NADPH. These are the three reactants that we need to power the Calvin cycle. What is the product produced? It's a sugar. Not any sugar, it doesn't say it here, but specifically the sugar glucose. Remember, this is an abbreviation. Glucose, and I said this is going to be our product. So this is where we get our second final product that I gave you in the initial formula. And it's not any sugar, it's glucose. However, we get a couple of other products right here. So these are also two other products from the Calvin cycle. We get the used form of what was once a chemically rich molecule, ADP plus P, and we get the oxidized form of an electron carrier. We get the oxidized form of the electron carrier, and we write it as NADP plus to let a person know it is in its oxidized form. What do I mean by oxidized form? right? It's lost the electrons, okay? But it's for a good reason, okay? All right, so when you look at this PowerPoint, remember I first focus on light reaction, then I focus on Calvin cycle, but there's a little overlap here, all right? This is what we're going to visit in detail as we go. Okay. Starting with the light reaction, the first of two stages. Where does it occur? It occurs in the chloroplast thylakoid, specifically the membrane, and I'll show you a figure. So they're showing you these little discs, but I'll show you a magnified image that they gave it, that the publishing company gave us, representing the membrane of the thylakoid, showing you a lot more detail. The purpose of the light reaction: to produce chemical energy to power the Calvin cycle, electrons and ATP, but I already said the electrons are actually being carried by this electron carrier. And how do we let a person know when it's carrying those electrons? We will write it this way to let you know it's in its form. If we want you to know it's not carrying electrons, we rewrite it this way. Okay. How many electrons can it carry total? Well, just like the other ones we talked about for cellular respiration, this electron carrier can carry 2 electrons. Okay. So this right here is the purpose, but these electrons are being carried by this guy here. So it shows you here. And we do get a waste product, O2. So this is what I'm talking about. This is a formula I gave you for photosynthesis. Your textbook gives you 2 formulas. The one I'm giving you is the reduced form, and that's the one that's most popular and the one that serves its purpose. Okay. What does this represent, everybody? What is that supposed to represent that I put a bracket underneath? Right, glucose. I put that down because sometimes without glucose, sometimes they'll give you, it's like a formula. The catch is, normally we write out glucose. Remember, again, the 3-letter abbreviation. Normally we'll write glucose because all monosaccharides have this formula, but sometimes they'll do this. Okay, so listen carefully. From this formula, what I've done is I've extracted what we need for the light reaction. We need H2O and we need light. And I simplified it. I didn't put 6 molecules. I'm just saying for the light reaction, we need these 2. And then from the product side, I extract O2. Technically, it's 6 molecules. So over here, it should be 6 of water, and over here, it should be 6 of O2. Notice there's a 1-to-1 relationship. 6 H2O going in, 6 O2 coming out. I'll explain this in a second because it has to do with where the electrons are coming from that this guy's carrying. But if you notice, for the light reaction, what we don't have in the original formula are products that happen in between. They're not final products, so you don't see them. This formula is telling you these are the final products. That's it. This is what's coming out of the chloroplast. But if you're just talking the light reaction, there are actually 3 products. One is a final, and then these 2 here that are required to power the Calvin cycle. So that's just another way of rewriting it out to help you. All right, then what you find out, or what my students have found out in the past, is what I've covered up to now is the easy part, okay? Because we're going to go into detail now. If you look here, what does this right here, this area look like? If I were to ask you, describe what this structure is, what would you tell me that I'm circling with the finger here? Can't circle it forever. Yeah, phospholipid bilayer or cell membrane, either one, right? Phospholipid bilayer is more exact, but it looks like the cell membrane. But let me, excuse me, I'm sorry. But let me magnify this. They're actually showing you the membrane of a what? Thylakoid. One of those green discs. Okay? And what they're trying to show you is what you find inserted into the membrane of a thylakoid. Okay? It's a molecular complex called a photosystem. So I'm going to jump back and forth. Sorry about that burp. I don't know where that came from. Okay, so if you look here, down here, this is in your notes, and I edited it real quick this morning. The best way I can describe what this structure is, a photosystem here, is down here. In your notes, I have it right next to photosystem. It's a solar-powered molecular complex consisting of a reaction center, which they show you here, light harvesting complexes, which they show you here. Okay. Now, why do I call it solar powered? Because down here, what molecules do you find in the reaction centers and the light harvesting complex? Chlorophyll. So this is where the solar power is coming from. The molecular complex is it's made up of proteins and light absorbing pigments. There's other molecules involved. It even has what we call a primary electron acceptor, which I'll mention. It's not like O2, the final electron acceptor. This one is more of a molecule that passes on electrons to ensure that in the long run they end up with NADPH. So going to this other figure and focusing on these two, if you look, the reaction center here is in the middle. It consists of chlorophyll, which they show you here, plus a primary electron acceptor, which they show you here with the box. It's surrounded by multiple light harvesting complexes. So notice they're saying multiple here, telling you it's plural. Multiple light harvesting complexes. So here they're showing you 1, 2, 3. What do you find forming this? Well, it's proteins, but also chlorophyll and some other accessory pigments. What do I want you to focus on for these two parts that make up a photosystem? The chlorophyll in each, and then in addition to that, the primary electron acceptor for the reaction center, because I'll mention that briefly. So using this other figure, in the membrane of the thylakoid, which looks like the cell membrane, okay, in the membrane of the thylakoid, we have these molecular complexes called photosystems, and I call them solar powered because these little green beads that you find throughout the photosystem represent chlorophyll, okay? But when you look at the anatomy of the photosystem, yes, chlorophyll is found throughout the photosystem, but they discovered it actually consists of two parts. The reaction center, where we have chlorophyll plus a primary electron acceptor, and it's surrounded by multiple light harvesting complexes, where the main part are the chlorophyll molecules. Okay, over here it says pigment molecules, but they're talking chlorophyll molecules. Okay, wrong way, hold on. Now, or actually, let me read this real quick. So here it says photosystems in chloroplast thylakoids capture light energy, capture light to excite electrons. This has to do with the chlorophyll molecules. Now, what they found is that there's not one type of photosystem, there's two types. I know the primary electron acceptors are part of the difference, but I haven't looked up where the other differences occur. So what I want you to know is, one, there's two photosystems, photosystem 1 and photosystem 2. These photosystems sometimes go by the abbreviation PS1 and simply PS2. Of course, when I'm writing it, it's not that simple, but if we were meeting in person, it'd be a lot easier. So what they discovered is that for the light reaction to occur, okay, it requires photosystems. There are two types of photosystems, simply known as photosystem 1 or PS1 and photosystem 2, PS2. In general, they both have a reaction center with chlorophyll and a primary electron acceptor, surrounded by multiple light harvesting complexes where the key molecule is chlorophyll plus some other molecules such as proteins. Now, what researchers discovered is that for these photosystems, you will find electrons flowing through. Okay, and if you notice here, they're showing you this. So what they found is electrons flow through the photosystems. However, electrons can take one of two paths when flowing through these photosystems that are associated with the first stage. So either electrons are going to flow in a manner described as linear electron flow. Okay, so when electrons are flowing through these photosystems, if they're flowing in a manner in a path that they call a linear electron flow, that means they're going to flow through both photosystems. But what they discovered is sometimes, for some reasons not understood, sometimes when electrons flow through the photosystem, they actually only flow through one photosystem. And then they'll flow out and they'll come back to that same photosystem, and they'll flow out and they'll come back. So whenever electrons take that path, they call it cyclic electron flow. The most common, the one we're going to go into detail on, is linear. So here they're showing you the two photosystems. The most common path that electrons take when flowing through photosystems of the light reaction is linear. But notice, the order in which the electrons flow through the photosystems is not in the manner that they're numbered. Now, I haven't looked that up. I'm just assuming there was a reason for it. It might have to do with cyclic electron flow, but I'm not sure. But we're going to talk about in more detail how light particles, along with chlorophyll, remember light particles are called photons, along with chlorophyll molecules, and an enzyme, I haven't been able to find the exact name of the enzyme, but an enzyme will strip electrons from H2O, and then with the help of these light particles, the kinetic energy from them being transferred to the chlorophyll molecules, which now have energy themselves, will help electrons flow through photosystem 2, through a complex of proteins known as the electron transport, chain, and then into photosystem 1, where more molecules of chlorophyll are being excited by photons, which they don't show you. They show you up here, ensuring that the electrons eventually flow to our electron carrier in its reduced form in ADPH. And as the electrons are flowing through the ETC, which bridges the gap between PS2 and PS1, I'll show you more detail. The ETC, just like in cellular respiration, creates a proton concentration gradient, which gives an enzyme called ATP synthase the energy it needs to perform chemiosmosis, which gives us ATP. Now, I'm not going to go into as much detail here, but I'll show you what I'm talking about here, how this middle part for linear electron flow, it falls right in line, or close, 99% in line with what we talked about for cellular respiration. That's the third stage, oxidative phosphorylation. Cyclic electron flow. Now, they don't understand why it occurs. They do understand the purpose of it, though, to produce excess ATP. Now, scientists don't understand that because plants perform cellular respiration. They're making ATP already. Why does it need to be made during photosynthesis? They're not sure, but they know it happens. For some reason, as electrons flow, and it doesn't say ETC here, but where it says cytochrome complex, they mean electron transport chain. For some reason, sometimes electrons that flow out of photosystem through the electron transport chain will flow into photosystem 1, and instead of being given to the electron carrier in its reduced form, NADPH, the primary electron acceptor of PS1 will send them back to the ETC, and they'll just keep handing them back and forth. But when this is happening, the ETC keeps that concentration gradient formed. Thus, ATP synthase synthesizes extra ATP. But nothing's produced for the Calvin cycle, so that's what they don't understand. This is called cyclic electron flow. What I want you to know for the test is the direction the electrons are flowing between the ETC and PS1. The purpose, produce excess ATP. Why? We don't know. So what I'm going to focus on, though, is the most common path that electrons take. It's called linear electron flow. Now, please remember, whether we're talking about a path taken by electrons called linear electron flow or a path taken by electrons called cyclic electron flow, we are in the first stage. Our light reactions are reactions. Now, what I'm going to focus on now is linear electron flow. We're going to go into some detail here because that's the most common path taken by electrons here. Sometimes I like to go back just to kind of reset us as to where we're at. Hopefully that will help some. Now listen carefully. What I did here also is in your notes, I give you information on how linear electron flow works, how it works, and I try to give it to you in chronological order, step by step. But what I did is I copy and pasted some of these steps into the PowerPoint to help you follow along a little better and look at the visual, All right. So excuse that noise if you heard that. Have any of y'all been having trouble with Office 365 by chance? I don't want to jinx you, so knock on wood if you say no, or don't say anything. Well, we've been having all kinds of trouble. I forgot to tell y'all, we might get cut off on Blackboard too, so let me hurry up here. All kinds of problems. All right, so linear electron flow, how does it work? Well, this is most of the process. The only thing missing is the electron carrier, okay? But when I go over linear electron flow, I'm going to start with here at PS2, okay? Then I'm going to go over this and how that leads to this. As electrons continue to flow into PS1, and what they don't show you is eventually these electrons will flow out of PS1, two at a time, to our electron carrier, if I don't screw this up, NADPH. Oh, almost messed up. It's pretty messed up, I mean, but you know, it could be worse. Y'all have seen that already. To NADPH. So, and I'll show you with more detail towards the end. So this is what we're going to go over. So we're going to begin over here on the side of PS2. So again, I copy and pasted some of the information. So in your notes, I have first, step by step. Linear electron flow first begins with photons that photons that excite chlorophyll molecules. So they show you here. Here's our first of three reactants. Photons, which are light particles, they're in constant movement, therefore it's a type of kinetic energy. Chlorophyll molecules have the ability to harness or redirect that kinetic energy. Well, basically what's happening is these light particles called photons, they're a type of kinetic energy, energy of movement, and when they come in contact with these molecules of chlorophyll, that transfers this kinetic energy over to the chlorophyll molecules. Now kinetic energy has been transferred to them. Now they're excited, they're moving. Okay, now why do we want these chlorophyll molecules being excited and moving or transferring the kinetic energy from light? Because as the light's coming in, so is H2O. Now, as I said, I couldn't find it. There's an enzyme, okay, that while these chlorophyll molecules are being excited, okay, by light, or while this kinetic energy is being transferred to the chlorophyll molecules, there's an enzyme that will take a molecule of H2O and strip 2 electrons from it. So if you look here, okay, going back, first photons excite chlorophyll molecules, which in turn help electrons flow through PS2. So the job of the chlorophyll molecules is to help electrons flow from the light harvesting complex and to help these electrons continue to flow through the reaction center to our primary electron acceptor. That's where these electrons are flowing. So that's the role of chlorophyll, here and all out here. Okay. Light energy. See, kinetic energy, the energy of movement. Photon particles are moving. They strike the chlorophyll molecules. That transfers kinetic energy to them. They're excited, they're moving, and they're making sure electrons move out of the light harvesting complex into the reaction center towards the primary electron acceptor. But where are these electrons coming from? Because I didn't finish. Well, these electrons are coming from H2O. H2O is our second of 3 reactants we need for photosynthesis, but specifically H2O is a substrate. It's acted on by an enzyme, and it is oxidized. It loses 2 electrons. When H2O loses those 2 electrons, it's converted to O2. That's one of 2 final products for photosynthesis. The O2 is coming from H2O. Do you recall I said a 1-to-1 relationship? We need 6 molecules of H2O, but we get 6 molecules of O2. It's because each molecule of H2O is losing 2 electrons. It's being oxidized and broken down to 1 molecule of O2. The electrons that chlorophyll molecules are ensuring will flow through the light harvesting complex and into the primary electron acceptor, they've come from H2O. Now, once these electrons derive from H2O, okay, with the help of chlorophyll, flow and reach the primary electron acceptor in the reaction center, okay, and it says here, it says it will flow through PS2, so I've got it better in your notes, okay, so the electrons will flow through PS2 to the electron transport chain. Do you recall the abbreviation for the electron transport chain? Just the ETC. Just like with cellular respiration, the electron transport chain is made up okay, a protein complex. The job of the protein complex, I tell you down here. So once the electrons flow to the primary electron acceptor, they will flow out of PS2 to the ETC, which they show you right here. And it says here, second, the flow of electrons through the electron transport chain powers the chain as a proton pump to create a proton concentration gradient across the thylakoid membrane. Well, that's synonymous to what we talked about for cellular respiration. And what's the purpose of creating this proton concentration gradient? So that protons will diffuse through ATP synthase, which they're not showing you here. So imagine the ATP synthase is represented by the number 5, okay? So if it helps any. Imagine the ATP synthase is represented by the number 5. Protons will diffuse through ATP synthase, giving it the energy it needs to synthesize ATP, okay? The catch is this: unlike cellular respiration, as the electrons flow through the ETC, giving it the ability to create that proton concentrating gradient, which allows ATP synthase to synthesize ATP, there's no O2 as a final electron acceptor. Instead, electrons flowing through the ETC, okay, will flow directly into PS1. So third, electrons flow from the electron transport chain, and then they will flow into and through PS1, where more molecules of chlorophyll are being excited, okay, or are absorbing and capturing and redirecting light energy and putting it to work to where the chlorophyll molecules are excited. Now they're moving, and they ensure the electrons entering from the ETC into PS1, the light harvesting complex. The chlorophyll molecules ensure that they move into the reaction center of PS1 and then to the primary electron acceptor of the reaction center of PS1, okay, with the help of these light excited chlorophyll molecules. So again, the best way I can describe it is playing pool. The cue ball is light, the chlorophyll molecules are the 8-ball. When you hit the cue ball, okay, the cue ball moves. That's kinetic energy. When it strikes the 8-ball, which would be the chlorophyll molecules, that transfers kinetic energy over to the 8-ball. Well, light energy, kinetic energy, that energy is transferred over to chlorophyll molecules, which in turn are excited, or now we have kinetic energy flowing through them. Once the electrons reach the primary electron acceptor of the reaction center of PS1, an enzyme, I'm not going to test you on this, but there is an enzyme. It shows you right here. I will not test you on the name. But then what will happen, all I want you to know from here, what will happen is in the primary electron acceptor, okay, will ensure that the electrons are transferred to the electron carrier, NADP+, that enters in an oxidized form. Once that electron carrier picks up 2 electrons, it exits, okay, it exits the light reaction, or it in a reduced form, NADPH. So you can see here they're showing you 2 electrons, which will be transferred to this oxidized electron carrier, which in turn reduces the electron carrier. Okay, so we have that now. Now, this is the entirety of linear electron flow. This is everything. Okay, now as I said before, we need light, we need water. Okay, what are the products that we get? We get the final waste product, O2, but we also get ATP, and we get our electrons carried by the electron carrier in NADPH. Of those 3 products, okay, this product and this product will be transferred, and I'm going to abbreviate this, are going to be transferred to the Calvin cycle. So I'm just going to abbreviate it CC. Your next exam is all multiple choice, okay, everybody? So just make sure you can recognize. So we have 3 products. One is a final product that I gave you in the original formula for photosynthesis. The other 2 are products of the light reaction also, but they're not final products. They're taken over to the Calvin cycle. These are our sources of energy. Okay, listen carefully before I show you the next figure. I'm going to go back here. I might have to go a ways back. Yeah, I'm going to have to go a ways back. Okay. These little discs right here, these little green discs, I'm going to show you a figure where what they've done is they've taken, let's say this up here. I'm going to show you a figure where what they've done is they've just taken a piece of the thylakoid membrane, okay, like they did here, except they're going to show you a wider view of the thylakoid membrane, where you're not only going to see a photosystem, you're going to see both photosystems. You're going to see the ETC, you're going to see ATP synthase, okay? You're going to see, and the proton concentration gradient. You're going to see all of it. So in the end, they have you, the way I approach this is giving it to you in bits, and it is tough to follow. But then take all those steps we went over for linear electron flow and apply it here. This says this is a thylakoid space where we have a high proton concentration. That means this phospholipid bilayer here must be the thylakoid membrane. Okay, now insert it into the thylakoid membrane. There it is, they're showing you PS2. Okay, they're showing you PS1. I don't know why they didn't say it, but they're showing you this, which they should have just said electron transport chain. So wherever you see cytochrome complex in between PS2 and PS1, just rewrite it as electron transport chain or ETC. Okay, and then if you notice also, they show you this. So both PS2, PS1, the ETC, and ATP synthase, where do you find them? Inserted into the thylakoid membrane. So just to repeat everything for linear electron flow, water, which is actually in the thylakoid space, will enter photosystem or PS2 at the same time light energy is exciting the chlorophyll molecules. An enzyme will oxidize or strip two electrons from H2O, converting it to O2. The electrons will flow through the light harvesting complex into the reaction center with the help of chlorophyll molecules in PS2. The electrons will flow out of PS2 through the ETC. As the electrons depicted with the yellow line are flowing through the ETC, that allows the ETC, okay, to pump electrons from the stroma, where there's a low concentration of protons, into the thylakoid space where it's a high concentration. As these electrons flow out of the ETC, they will flow into PS1 at the same time. This proton concentration gradient results in protons wanting to diffuse from a high concentration to a low concentration. And where do they find that they can? They can flow through the enzyme ATP synthase. So electron transport and chemiosmosis are occurring, but if I ask you specifically for those two terms, it'll only be in regards to cellular respiration. I'm just explaining the bigger picture here. Protons will flow down their concentration gradient. This gives this enzyme ATP synthase the energy it needs to do work in the presence of a proton concentration gradient, thus synthesizing ATP. While at the same time, simultaneously, electrons flowing out of the ETC flow into the light harvesting complex of PS1 now, where more chlorophyll molecules are absorbing light or being excited, ensuring electrons flow through the light harvesting complex into the reaction center to the primary electron acceptor of PS1, out of the primary electron acceptor, and eventually to the electron carrier NADP+, which gains 2 electrons and is reduced to NADPH. And then they show you where is this electron carrier being reduced and where is ATP synthase synthesizing ATP? On the side of the thylakoid membrane that is in the stroma. Now, why is it important for this electron carrier to be located in the stroma and for ATP to be synthesized out here in the direction of the stroma, even despite this being inserted into the membrane? It's because the Calvin cycle occurs in the stroma, everybody. This is where the Calvin cycle occurs. And I put down here both ATP and ADPH are transported through the Calvin cycle. Okay? So when you go over everything, in the end, you want to look at this figure, and you want to start over here. You want to read what they have, but when you get started, you want to go this way, towards this side. Okay, stage 2. I'm just going to mention this briefly, and then I'll cut it short so I can finish up on Wednesday. But if stage 2 will be what we'll pick up with on a Wednesday. We will begin with the Calvin cycle. And in order for the Calvin cycle to occur, we need the reactant, which is actually a substrate, carbon dioxide, and we need ATP and NADPH. I inserted this formula into the notes this morning, so forgive me if you have saved a copy before then. Go back, and I inserted this formula back in. And we will talk about, okay, how during the Calvin cycle, it's broken up into 3 phases. I will give you the name of one enzyme for sure. It's called rubisco. The reason why I give you the name of that enzyme is because it is considered the enzyme of most importance because of what it does. Rubisco performs a process called carbon fixation, and that is phase 1 of the Calvin cycle, as we'll talk about on Wednesday. This enzyme is popular because of its ability to perform this process called carbon fixation. And as I'll talk to y'all next week, I'll tell you carbon fixation, I'll tell you again on Wednesday, sorry, not next week, but again on Wednesday, that carbon fixation is a process whereby atoms of carbon in CO2 are covalently bonded together, creating a carbon backbone. Everybody, what type of molecules have carbon backbones? In general, what do we call that group of molecules that all have a carbon backbone? We had a whole chapter devoted. Right. Well, I said the purpose of photosynthesis is to produce glucose. Is glucose an organic compound? Yes or no? Is glucose an organic compound? Yeah. Well, this is still a major area of interest to scientists, because what they found is in the process of photosynthesis, the molecules of glucose that are produced by photosynthesis, the atoms of carbon come from CO2, the inorganic compound. Why would that be of such importance to the scientific community as a whole, especially in regards to global warming? Don, maybe I should ask you that test question. Why would scientists be so interested in this in relation to global warming? What do they believe is the cause of global warming? The primary cause. Boy, everybody's on social media and stuff and over, right? Carbon dioxide, increasing CO2s. It's a greenhouse gas, right? Right. So the thought process is this: CO2 levels are already high. So agreements like the Paris Agreement, it's only addressing not, trying to slow down the rate at which these CO2 levels are continuing to increase. But there are scientists that say, well, even if we stop increasing the levels of CO2, they're high. We need to fix that. So there are those scientists looking into what they call bioreactors. Okay. These are basically like your water furnace, the electrical wiring in your house. They're trying to modify organisms, create GMO organisms that can take CO2, perform carbon fixation, but instead of producing glucose, they're hoping they'll produce some type of fuel source like ethanol or something to power your house maybe, or part of your house. Okay, thereby reducing the amount of CO2 in the air. So it comes from areas like this. I'm not going to test you on that, but I'm just pointing out because I have students, oh great, photosynthesis. Well, if you're narrow-minded, yeah. But if you're very young, you've got to pay attention to what's going on around you. This is an area of interest, okay? All right, I'm going to stop the recording there.